Gotten through a lot of material this minute, or at least a lot of slides. Huh. I'll give you a couple minutes. Go ahead and start putting our axes on and some of the key points in our action potential that we want to be aware of. Hopefully everybody's drawing started out like this. All right. Wants to jog my memory about what kind of a line I will be drawing for depolarization. Going up, and from where specifically? From 70 up to 100. Exactly. So up to 55 to our threshold, and then up to positive 30. So who wants to tell me why this is happening? What ion are we thinking about? Sodium here. So we're thinking about the sodium rushing in through those voltage gated channels. This is our sodium coming in. All right, what's next with my curve? It's our second phase. What does repolarization mean? Yeah, exactly. So we had our sodium and they are closing. I'm going to cross them out. We're inactivating them. And now we're letting our potassium flow. It likes to flow out. So we are repolarizing, which means we're going back down, which just leaves us with hyperpolarization. So what's a hyperpolarization? Perfect, yeah. So when we go below -70, usually gets to like -85, that's our hyperpolarization. And that's still due to our potassium leaving. We're slowly cutting off that potassium permeability. And then our ATP pump would kick back in and we'd restabilize at -70. So this very stereotypical curve, that is an action potential. That whole thing will always look roughly like that, right? Some big spike and then a dip. We're always looking for that when we're looking for an action potential. Pointing out that we're always looking for the same exact curve for an action potential because we have this principle called the all or none principle. All that means is that either we have an action potential or we don't. We don't have like really big action potentials. We don't have really small action potential. An action potential is an action potential is an action potential. They don't vary. They're always that same shape. They're always going between those same membrane potentials from -70 to -55, up to +30, dropping down probably to -85, and then restabilizing. The only place we might expect to see any variation there is in that hyperpolarization when they drop below 85. We'll see why there's maybe a little bit of wiggle room there, but there's nothing that's going to differ in that depolarization that spikes at +30. Fundamentally, that's all the all or none principle is. Depends on this threshold, right? Our threshold is -55. So that's our minimum. We need to get graded potentials somehow adding up to -55 in order to induce our action potential to kick it off. We call it regenerative because we have kind of positive feedback, some sodium channels opening, more depolarization, more sodium channels open, and then they reset to their original state. It happens no matter what, so long as we get to -55. And we will see sometimes people refer to sub-threshold depolarizations or supra-threshold depolarizations or just threshold depolarizations. When they do that, they're referring to those like summed graded potentials. Those aren't about the action potential. Those are about the events leading up to it, about how we got to that -55 threshold. So if we just have a threshold depolarization, that means we get to -55, boom, action potential goes off, we're good. A sub-threshold depolarization means that we do have an excitatory graded potential, meaning our cell gets a little more positive than -70, but we don't get to -55. So say we get just -60. That would be a sub-threshold depolarization. It still counts as a depolarization because all depolarization is referring to is the fact that we're getting more positive from -70. But it's not this wave of depolarization that we have in our action potential. And if we don't get to -55, no action potential, we're done. So that's our sub-threshold depolarization, which is a way we might describe a graded potential, or we might say that we're describing a response to a stimulus, but we know that a graded potential is a response to a stimulus, basically. Suprathreshold depolarization could happen. You could have lots of stimuli coming in or really strong stimuli. And say those graded potentials add up together and they get us to -40 or something. That would be a suprathreshold depolarization. And that can happen. But it doesn't make your action potential bigger. It still just kicks off an action potential. We're going to spike to the same +30 as always. And that's really why we bother talking about the all or none principle, is because our graded potentials do vary in magnitude. They can be bigger or smaller. We could describe them as threshold, sub-threshold, or suprathreshold depolarization, but none of those terms would make sense to describe an action potential because it just exists or doesn't exist. And that's it. The only thing that you will see potentially varying with action potentials is how close together they are. So in this image, they're not focused on the full shape of the action potential. So they're just showing you that spike to positive 30. So they're just kind of waving their hands about the different phases. So you will never see one that goes like this, and then one that goes like that, right? Those would not be action potentials, right? Because they're not the same thing, this depolarization, positive 30. The other thing about this all or none principle is that it doesn't affect how wide these action potentials are, which is to say that a single action potential is going to take the same amount of time in this one area of the membrane. So we're going to talk about propagation of action potentials and how they pass down the length of a nerve or down the length of a neuron. That's not what we're talking about here. When we're looking at this little curve, we're kind of just looking at what's happening in one little segment of the cell, in one little segment of the axon. So that segment wouldn't like hold on to the action potential for longer or hold on to it for shorter. Always going to be in that area for the same amount of time, regardless of the stimulus. So we're not going to change the height. We're not going to change the width. The only thing we could potentially change about action potentials is how close together they are. Like we could have potentially like another one in the middle here or something, right? So this would be a bunch of action potentials firing off closer together. There's a limit on how close they can get, and we'll talk about why that is. But that's the only thing you should expect to see varying when we're talking about these action potentials, these nervous impulses. So here we just see more images about this, right? So here we have stimuli, subthreshold stimuli, graded potentials of different sizes. So we can see the graded potentials here. We have a small one, we have a bigger one. Neither of them were big enough to kick off an action potential, so they just fade away because our graded potentials are decremental, they decay, they fade. If we get to threshold, we kick off an action potential. And if we even have a super threshold stimulus, so like a really big one, we still just kick off that same action potential. Absolutely nothing different about it. So the only difference would be in that initial stimulus and in the graded potential, not in the action potential. So let's take a look. Can have that. Do they really?
But no, by all means, go ahead.
I am going to skip the yes. And we can hear the robot tell us about action potentials now. Oh, am I doing that? Nope, they're doing that. Okay.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A cluster look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative key events occur. The inactivation gates of voltage-gated sodium channels close and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the outside, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of -70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through weak channels only, and the membrane potential returns to the resting state of -70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the outside, repolarizing to +30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the outside, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of -70 millivolts.
Okay. Questions about that graph, that picture of the action potential, or the all or nothing principle? And now I'm going to go through it in flowchart form. Modality. Okay. This flowchart is from your book. So remember, it's going to have those different colors. Depending on green, it's going to tell us what takes things off. And we're going to have responses. So when we see little up arrows next to the text, that means that we're increasing something. Little down arrows means we're decreasing. We'll have little dashed lines for positive and negative feedback loops. In a positive feedback loop, you wouldn't have to flip the direction of those arrows. But in a negative feedback loop, you're suppressing. So any arrows you see would flip over. So our initial stimulus here is just a threshold stimulus. So we got some signal from somewhere out in the body that we should be firing off a nervous response, potentially. So our threshold stimulus is going to start to create a depolarization of the message. So depolarization means it's coming up, right? Our membrane potential is starting to climb. Since we have specified that it is a threshold stimulus, right? We're going to reach negative 55. This means that we know we're going to open a bunch of sodium channels, and you know that this flowchart we're going through is eventually going to kick off an action potential. So the first thing that's happening here, we're increasing the number of open sodium channels, right? So you see that little up arrow. Starting to open sodium channels. We know that there's a really big electrochemical driving force for sodium to move into cells. So open sodium channels are how sodium would get into a cell. So when we have more open sodium channels, this means we also have greater membrane sodium permeability. So permeability is kind of just about how many doors are open in that membrane. So those two statements are basically the same thing. They're inextricably linked together. So we got a bunch of doors open. We know we have this big electrochemical driving force, which means sodium is flowing into our cell. Right? We're finally letting it do what it wanted to do the whole time. This means that our cell is going to get more positive. Sodium ions are cation, meaning they're positive ions carrying positive charges. We bring positive charges into our cell, starts out negative. Our cell is going to get less and less negative, and then eventually it's actually going to go into our positive range as sodium tries to get to its equilibrium potential of positive. We call this depolarization when we have this net positive charge in the cell. It's still depolarization, whether we get from -60 to -55, or whether we're going from 0 to 30, both of those refer to depolarization because they're both in the same direction. So it's about the direction here, not about the absolute number. This creates a positive feedback loop in our neurons because as we have these sodium ions flowing into the cell, this is opening more and more and more voltage-gated sodium channels. So more and more regions of the membrane, right, in this local region, are reaching that threshold that they need in order to open the channels. So note here we have another kind of split that's happening in this graph now. So another effect here, after we open the sodium channels, is that we have a delayed effect. So this delayed effect is still ultimately due to reaching that threshold stimulus and that initial depolarization. But it takes more time just due to the nature of what these proteins, these channels look like and how they work. So specifically the delayed effect here is that the sodium inactivation gates are closing. So those ones that swing close there. Remember that our sodium channel has that internal, like in the middle of the channel, activation gate. And then on the intracellular side, it has the inactivation gate. So this first loop that we went through had to do with the fact that the activation gate opened and the inactivation gate at rest is open. So we have two doors open. And now we have this delayed response. The inactivation gate closes. This causes the sodium permeability of this membrane to go down, decreases that membrane sodium permeability as we're starting to block off those sodium channels. So there should be a down arrow here. So if there's not on your page, go ahead and draw it on. This slows and ultimately stops sodium flowing into the cell. Once we have all these sodium channels closed, even though we still have some force for sodium to move into the cell, right? It's not at its equilibrium potential yet. Can't do it. It doesn't have any passage way. This means that our cell is no longer going to be going up towards sodium's equilibrium potential. But this is part of what is causing repolarization. We're not going to be increasing the positive charge in the cell anymore. The other half is going to come from a second reaction to our initial stimulus. And just to note here, I'm going to do our negative feedback loop first. So over on the side here, as we repolarize, that's like the opposite of depolarization. So that's a negative feedback loop. Right, so now we're closing this off. The other thing that's going on to help us repolarize is that we have a delayed effect on our potassium channels as well. So the delayed effect on the sodium channels is to close them. The delayed effect on the potassium channels is to open them. So they just chill out and don't do anything at the beginning when those sodium channels are opening. They're super slow. But once those sodium channels are starting to close, that's when our potassium channels are waking up and opening up. We see that increase in open potassium channels, which means we have an increase in membrane potassium permeability. Okay. Those two concepts should always mean the same thing. Basically, when we have open channels, we're more permeable to whatever the channel is for. So the other piece of repolarization is the potassium flow out of the cell. So the electrochemical driving force of potassium is in the opposite direction to sodium. So sodium always wants to come into the cell. So potassium always wants to leave the cell because it's trying to get the cell down to -94. So when we open up those extra potassium channels, potassium leaves the cell, and that's the part that's pulling down our action potential, repolarizing it. And that can continue a bit below our resting membrane potential of -70, and we get restabilized as the sodium potassium pump fixes our concentrations, basically. So these tables are basically for you to study from, and they're from your book, but we can use them briefly as a little review here. So remember, we've been talking for a couple days now about our different values of the membrane potential that we want to pay attention to. Rest is -70. Depolarization is this whole phase, -70 to +30. But we'd want to kind of remember that we need to get to -55 in order to complete this whole phase. Repolarization is just back down from +30 to -70. And the hyperpolarization, or the after hyperpolarization, what's happening kind of after the end of the action potential, is -70 to -85. So we want to be able to track during each of these phases, sodium flow into the cell, potassium flow out of the cell. So we want to know when in each of these phases that's happening. We also want to know the state of those channels, right? Which, if we know the flow, we know a lot about the state of the channel. Because if that ion is flowing, its channels are open. If that ion is not flowing, that's a good indicator that its channels are closed. Because in no part of the action potential do we reach the equilibrium potential for either of our key ions. Yeah. The leak channels, is that what you said?
Yeah, they're not the proteins, right? It's just through the.
So the leak channels are channels, so they actually are also proteins. Okay. They just don't have gates, and they're not really involved with the action potentials. Basically, there aren't enough of them is how I kind of think about it. So they're the ones that are responsible for getting us to -70, right? That balance between the sort of baseline permeability of potassium and sodium keeps us at -70. So they don't change at any point in the action potential. We also don't have very many of them in the axon, so they kind of get overwhelmed by what's going on with the voltage gated channel. Does that kind of make sense? Other questions about this stuff? Again, this is just more notes that you can use to jog your memory about all the stuff we've been talking about for the past several days. Specifically, this is a comparison between graded potentials and action potentials in case you're having any trouble kind of sorting out those two in your mind. Graded potentials just being those reactions at the beginning that we're trying to get ourselves to threshold. The action potential being really stereotypical, always the same, all or nothing curve with the three phases. And some checkpoint questions that you can mull over later as well. What I want to talk about next is our refractory period. The refractory period is like a pause that has to happen between our action potentials. When I said that our action potentials can only get so close together, this is the feature that we're really referring to. An action potential can't get taller, it can't get wider, right? So we can't peak above +30, and we're not going to have a really long action potential in any one location. They can increase in frequency, but only to a certain point. So we're going to talk about two things: the absolute refractory period and the relative refractory period. The absolute refractory period, you want to think of as like the absolutely not period, right? Absolutely, absolutely, there will not be another action potential during this period. The relative refractory period, as you might guess, probably not going to have an action potential here. You'd need a really strong stimulus to overcome it. Our threshold doesn't differ during the relative refractory period, but how far away we are from threshold does differ. Which is to say, basically, that the relative refractory period kind of is that after hyperpolarization, right? So you can imagine if you had to get from negative 55 to positive 55, sorry, negative 85 to negative 55, right? So we're still just trying to get to that threshold. It's farther away than negative 70 to negative 55. So it's not that you couldn't do it. You could potentially have a graded potential summed together that's strong. It's just probably not going to happen. It's going to be hard. So you'd have to have a very strong stimulus here. So the reason for these two periods differs. So the absolute refractory period is absolute because it has to do with the speed of the changes to the states of those voltage-gated sodium channels. There is absolutely nothing other than time that's going to make our sodium channel, our voltage-gated sodium channel, go back to its resting state. So during a certain period of time, that sodium channel is going to have the inactivation gate closed. And until it swings back open, it does not matter what we try to do to the activation gate in the middle. No sodium's going to get through. You just can't do it. So we have to have enough time for that inactivation gate to open back up to take us to the original state of the voltage-gated sodium channel. So that's our absolute refractory period. So it's happening during our rapid depolarization to positive 30, and also during this repolarization as we're coming back down. So remember, that's why we're repolarizing. That's like half of it, right? The reason we're not still climbing is because those inactivation gates swung closed. So they're closed during repolarization. So our main thing that we pay attention to, the thing we're going to always draw when we draw our action potential, is that spike and that drop, the depolarization and the repolarization. So during that whole spike and the drop back down, we're in our absolute refractory period. The relative refractory period is here in blue, right? So we see that after hyperpolarization, where we know we're getting below negative 70, so we would just have farther to go to negative 55. So we could do it, but we're probably not going to do it. Regardless, if we were to fire off an action potential in the relative refractory period, we wouldn't like subtract this from positive 30 and somehow have like a lower action potential. That's not going to happen. Okay. And if you have trouble with negative sentences, just like erase what I just said in your brain. So what I'm just saying is that if we got from here to negative 55, we'd still spike up to positive 30-ish, just the way we did with that initial action potential. It's still exactly the same. We're still obeying that all or nothing principle. So here, the graph on the left is changed just to show kind of the percent of channels that are inactivated versus open there. So we're kind of thinking about permeability here. And we have our absolute and relative refractory periods rephrased, but it's still just the absolute refractory period, all of depolarization, pretty much all of the repolarization phase. Can't make another action potential no matter what, because sodium gates are closed. Relative refractory period is mostly happening during that after hyperpolarization. You could consider the very last part of repolarization to be part of it if you wanted, but really associate it with the hyperpolarization more. So if we have a stronger stimulus during relative refractory periods, we could potentially have an action potential because some of those voltage-gated sodium channels have returned to their original state. Questions about refractory periods before we talk about propagation? Because understanding refractory periods is actually really important to understanding why there's a direction of impulse down there. There we go. Okay. So when we say propagation, all that means is like continuation. So what we've been talking about thus far is we've been like zooming in on a little bit of the membrane. We've been thinking about what is happening to a little chunk of the membrane over a period of time. So if negatives confuse you, close your ears for what I'm about to say next. But what you're not going to do is like overlay the action potential in your mind on the whole axon. That's not what's going on. It's not telling you what's happening at different places. It's telling you what's happening in just one spot. So like if we measured just here, we would see that spike. So what happens when we conduct a nervous impulse is we see that spike happen first here, and then we have the repolarization here and going back to rest. But then we see a spike here, we see a spike here, we see a spike here, and so on. That's what that like white light traveling down the axon in the video was. That was our depolarization event. So we first had one part of the axon depolarized, and the next, and the next, and the next. That's what propagation means, or we might call it conduction. So when we started out, way back when talking about membrane potential, we had our fake little circular cell. Now we've been talking about the fact that we can have local changes, and now that's really important as we think about our long skinny axon. Right? Our little local chunks of the axon make a difference. So we're going to expect those voltage-gated sodium channels to like be doing the wave down the axon. Actually, so yeah, like propagation down the axon really is like a crowd doing the wave, right? Hands up being the depolarization. The action potential happening at one section of the membrane is the depolarization that gets the next section of the membrane to threshold and kicks off the action potential in that location. Okay. So it's basically the spread of the charges that's like diffusion to neighboring areas that kicks off their own action potentials. So an action potential is going to be produced down the entire length of the axon at every patch of the membrane. Okay. And right now we're talking about an unmyelinated neuron. I want to be specific because we will see that myelination, which is like a wrapping around the axon and some other sort of helper cells, will make a difference, and we'll tweak the picture a little. But in an unmyelinated neuron, which is our basic type of neuron that we usually see, we have to trigger every single part of the membrane. So we're starting at the axon hillock, right? The axon hillock being that hinged off point, right? So that's what we see here. Okay. And the depolarization, that wave of depolarization, that action potential, is going to spread all the way down to our axon terminal. It is always going to go in that direction. It is not going to come back to its own cell body. It's only going in the one direction from the axon hillock out to the ends of the axon. Now, conduction in an unmyelinated neuron is pretty slow. at least relative to our myelinated neurons that we're going to see. This is because we have to fire off every patch of the membrane. So each little section of the membrane has to fire off, so that takes time as it spreads out. Remember, that last bullet point is just still reminding you of this all or nothing principle. So as we move down the axon, we're always seeing that spike to positive 30. We're specifying this here because graded potential, right, the reason we couldn't send a graded potential down to the end of the axon terminal is because they what with distance? What happens to them? Yeah, exactly. They dissipate, they fade, they're detrimental. So graded potential wouldn't be able to reach that far because it would get weaker, but an action potential triggers every single piece of the membrane. It stays the same strength all the way down. We're going to look at each of these pieces of the picture in turn, so I'll start clicking through. So here we see our same old neuron that we've been drawing, or that I've been attempting to draw. So we see that soma, the cell body. We see that axon hillock, the axon itself, and our axon terminals. So we're going to be conducting this neuron that way to the right. Yes, I can tell right from left. Awesome. If we zoom in on the axon hillock and this first patch of membrane, at rest, we have more anions inside. We have more cations outside. Right, so we see these negative charges accumulating along the intracellular portion of the membrane. These positive charges are on the external, the extracellular fluid portion of the membrane. So this is just what's normally happening at rest. This is our normal average neuronal cell at -70. As we start to fire off the action potential, we're going to start to trigger portions of the membrane. In this image, we can see site A currently undergoing that depolarization effect. So what are those positive cations that we see inside of site A? What are those representing? Yeah, exactly, sodium. So that's our sodium moving in. And those arrows moving to the side are telling you that sodium ions, once they move in, they're not bound onto anything. So they're kind of spreading to the sides. So we're going to see that that is what's going to be triggering site B next. But why do you think these sodium ions that are passing back towards the axon hillock, why aren't they triggering this section of the membrane? Why don't we trigger the middle and then have direction in both ways? Yeah, exactly. So the refractory period, the fact that those inactivation gates are swung closed in the part of the axon that has just fired, means that even though our sodium ions, like our sodium ions don't know what way to go, right? They just spread out. But it doesn't matter to the section that has just fired because it can't fire off again this soon. So that little depolarization that may be happening in the site that just fired doesn't trigger a new action potential. It does, however, trigger an action potential in site B. So as those sodiums that got into site A spread out, they raise site B up to -55. So now site B has an action potential. So we can see here site A, right, is now listed as being in the refractory state. Right? It's in the refractory state, meaning that even though some of those sodium ions drift towards it, doesn't do anything, can't do anything. So this is why the refractory state is really important. Like it is important that you kind of regulate the speed of action potential, but if we had no refractory state, we'd have a huge mess in our neurons, right? Like charges would be going any which way. We wouldn't be able to have impulses like out to the body. We just have a mess of electrical activity all over the place. So then we can see that as site B triggers, its positive ions, its sodium ions start to spread out. They trigger site C. And we can see that as we're moving down, right now site B is in the refractory state, but site A repolarized and it's back to resting now. So now we could start to trigger a second action potential, right? So here is how we might trigger another one, right? So it would be separate, it would have its own curve and we'd see it as a separate impulse if we were measuring here, if we were measuring at the axon terminal. But things aren't in the refractory state forever, they're just there for a time. So we're at time, so this is a great place to leave off. So when we meet next time, we will talk about how a myelinated neuron is a little different